This is your lecturer for evolutionary biology, John Bukenya, from the Institute of Open Distance and E-Learning at Makerere University. I would like to welcome you, dear students of evolutionary biology, to this session of an introduction to evolutionary biology. And I would like to share with you some slides, about 20 slides that are introducing this subject so that you know what it is all about. We are going to begin by looking at the term organic evolution. The term evolution, of course, you are aware, refers to change in life forms that occur, that take place as time changes. They occur with changes in time. But we will focus on the term organic evolution to mean the theory that the more recent types of plants and animals have their origins in other pre-existing forms, and that the distinguishable differences between ancestors and descendants are due to modifications in successive generations. To simplify this term, the theory of organic evolution is all about comparing the present day organisms that we see on Earth and relating them with their ancestors that lived in the past. Therefore, this theory looks at the different types of plants and animals that live today, and looking at them from an angle of the similarities and differences they might be having with those that lived in the past, their ancestors, and that the differences between ancestors and descendants are due to modifications in successive generations. Modifications in morphology, modification in physiology, in anatomy, in the biochemistry, in the behavior, and other aspects. Charles Darwin did not invent the idea of organic evolution. Generations preceding him entertained that notion that organic evolution actually takes place. An example is the French zoologist Jean-Baptiste de Lamarck, and this was a pioneer in invertebrate paleontology. And another one is Erasmus Darwin, the grandfather of Charles Darwin. But it was until Darwin's time, however, that the idea got greater currency, wider currency, because the earlier workers such as Erasmus Darwin and the French zoologist Jean-Baptiste de Lamarck lacked important data. They lacked important data to support the theory that organic evolution actually takes place. Charles Darwin's contribution was to propose a mechanism known as natural selection that explains how this change could take place. Darwin's theory, in his work, in the work of Charles Darwin, which is published as On the Origin of Species, first published in 1859. Darwin summarized his ideas as follows. The first idea in Darwin's theory is that populations of animals and plants produce progeny at such a rate that were they all to survive, they would increase rapidly year after year. The second idea is that spectacular progressive increases in population size do not in fact occur. Although most populations fluctuate year after year, they remain essentially constant over the longer term. So this second idea or observation from Darwin's theory is about constancy of species, while the first idea is about over-reproduction for any species. Now, idea number 3, a very real struggle for existence occurs in nature. Each individual must compete for food and must cope successfully with every facet of the environment, both physical, such as climate extremes, and biological, such as diseases and predators, in order to live, to produce progeny. This third idea is what we call struggle for existence. It's about individuals that have been produced competing for resources such as food and struggling to survive amidst the challenges that are physical and biological, so that they grow to the reproductive age and produce offspring or progeny. Another idea in Darwin's theory is that each individual differs virtually from others in its species. So this is the idea we call variation or genetic variation, also known as heritable variation. By Darwin's time, striking variation in domestic animals had already been produced by selective breeding. Darwin noted that species in nature had similar potential for modification. So Darwin observed that genetic variation occurs among individuals belonging to the same natural population. Darwin made a break with all previous suggestions on the subject and instead hypothesized that modifications are induced by the environment and are then passed on from generation to generation. He suggested that new characteristics arise from within an organism entirely by chance. We now know that these arise as genetic mutations. But by Darwin's time, the term genetic mutation had not yet been described. However, not all of these characteristics will have adaptive significance or survival value, and many may even be lethal. So this point means that whereas new characteristics can arise within an organism as a result of mutations, some of the new characteristics confer advantages to the organism, others confer disadvantages. So some are favorable variations, while others are unfavorable variations. But we are saying that some of the new characteristics enhance an individual's success in coping with the environment, an individual's survival. In other words, these are advantageous variations that lead to survival and growth of the individual to reproductive age. And this may even allow the organism to push beyond the previous environmental barriers. Others will be unsuccessful, and individuals with these modifications or variations would simply not survive to transmit them to their progeny or to the future generations. So this kind of differential survival and reproduction, where individuals with favorable variations or new characteristics of higher survival value survive and transmit their characteristics to their offspring or to the next generation, while those individuals without these favorable variations get eliminated or weeded out. This is a process termed as natural selection. Natural selection. To continue with the Darwinism theory, each individual, as we've said, or as we've just discussed, that while variations are produced in individuals, Darwin never explained the source of these variations. But another scientist, in fact, a geneticist known as Gregor Mendel, in 1865, demonstrated that organisms acquire traits through discrete units of heredity that we now know to be the genes. Much later, scientists in the 20th century, around 1930s, showed that genes are the ultimate source of variation within a population. That is to say, all variations arise through changes in genes, which changes we call genetic mutations. If a mutation enables an organism to survive or reproduce more effectively, that mutation tends to be preserved and spreads in the population through natural selection. Therefore, evolution depends on both natural selection and genetic mutations. Mutations provide abundant genetic variation, and natural selection sorts out the useful changes from the harmful or deleterious ones, those which can lead to death. Now, around 1953, two scientists, Francis Crick and James Watson, come up with another molecule, a polymer of nucleotides that is known as DNA, deoxyribonucleic acid. And I think you remember Crick and Watson's model of DNA. So with the discovery of this DNA, the study of evolution entered yet another phase, taking it to its most fundamental level. At this stage, we are now simplifying the issue of evolution at the most basic level, looking at DNA, because DNA is actually the carrier of the hereditary information that is transmitted from parents to offspring. Crick and Watson found that DNA contains genetic instructions used in the development and functioning of all known living organisms. Chemically, DNA is a long polymer of simple units called nucleotides, with a backbone made of sugars and phosphate atoms or phosphate groups joined by ester bonds. Still talking about the structure of DNA, attached to each sugar is one of the 4 types of organic bases or nitrogen bases. It is the sequence of these 4 bases, the nitrogen bases, along with the backbone that encodes information. And the main role of DNA is the long-term storage of information, the hereditary material, the hereditary information. Eukaryotic organisms like animals, plants, and fungi store their DNA inside the cell nucleus, while in prokaryotes, such as the bacteria, DNA is found in the cytoplasm. You remember from your knowledge of cell biology that eukaryotes have a true nucleus whose DNA is located in chromosomes inside the nucleus, separated from the cytoplasm by a porous or a perforated nuclear membrane. But whereas the nuclear membrane is present to separate the hereditary material or DNA from the cytoplasm, there is a mechanism which we call DNA transcription, by which the instructions in DNA inside the nucleus can be copied to form a messenger RNA that is small enough to diffuse out of the nucleus through the nuclear pores. So the messenger RNA can carry instructions from DNA to the cytoplasm in order to guide the process of protein synthesis, in order to guide the synthesis or the manufacture of proteins in the cell. So we are saying that within the cells, DNA is organized into structures called chromosomes, and the set of chromosomes within a cell make up a genome. These chromosomes are duplicated before cells divide in a process called DNA replication. DNA is the ultimate source of both change and continuity in evolution. The modification of DNA through occasional changes or rearrangement in the base sequences underlies the emergence of new traits and thus of new species. At the same time, all organisms use the molecular codes of the 4 DNA base sequences. This uniformity in the genetic code is powerful evidence for the interrelatedness of living things, suggesting that all organisms presently alive share a common ancestor that can be traced back to the origins of life on Earth. There is a new concept that we want to emphasize, that organic evolution, as described by Charles Darwin, can be discussed under 2 headings. The first heading is known as common descent. So we can have what we call the theory of common descent by Charles Darwin. And the second is known as descent with modification. So Charles Darwin's theory of organic evolution can be described under two headings. The first heading is common descent, which means common ancestry for all forms of life on Earth. Then the second one is descent with modification. So organic evolution by Darwin is emphasized as descent with modification, and also as common descent. According to this descent with modification, the present day complexities in animals and plants are as a result of evolution from simpler life forms that existed earlier through eventual changes. What we see today as complex structures in animals and plants, complex physiologies, complex behaviors, complex molecules, they have arisen from simple forms that existed earlier, but these changes from simpler to complex forms in the present day, animals and plants have resulted they have resulted from what we call modification. The structures in simpler life forms were gradually modified over a period of time to form the present day complex forms observed in plants and animals. Darwin's book on Origin of Species, published in 1859, was the earlier book or earlier paper to put forward the theory of evolution through natural selection. And this theory stated that the entities, the organisms, as a result of alterations in inherited behavioral and physical characteristics, undergo changes over a period of time. These changes allow species to better adapt to changing environments, helping in the survival of offspring. One of the best sustained theories has been the evolution by natural selection, backed by evidence from a range of scientific disciplines. So there are many evidences that support Darwin's theory of evolution by natural selection. And some of these evidences include the study of fossils, which we call paleontology, the study of fossils. Fossils being the remains of organisms that lived in the geological past. So the remains, the bones, the shells are all fossils. So fossil evidence is one of the evidences that supports that all present-day organisms have got a common descent. The descendants that we see today arose over time from their ancestors that lived in the past. Developmental biology also provides some evidence that there is a common ancestry for all life forms on Earth, and that there has been descent with modification. So developmental biology is basically concerned with embryology, the stages of development of organisms from the zygote up to the adult form. So those embryonic stages, if you compare those of different vertebrates, like reptiles, those of mammals, those of fish, those of amphibians, and those of birds, these embryos in the early days or early stages of life, early stages of development, they provide some evidence that the diverse groups of vertebrates living today must have had a common ancestor, and that they have evolved over time to the present day forms by or through descent with modification. Other evidences exist, such as comparative anatomy. For example, you can compare the pentadactyl limb in different groups of vertebrates. The pentadactyl limb in different groups of mammals, like in humans, in monkeys, and other groups. Somehow, that comparative anatomy, which also focuses on analogous structures, vestigial structures, and others, vestigial structures, analogous structures, homologous structures, all of those provide some evidence for either convergent evolution or divergent evolution, which we also call adaptive radiation. Another evidence can be obtained from biogeography, which is the geographical distribution of plants and animals in different biogeographic regions or continents. Further evidence is provided by genetics, by DNA sequencing, and so on, as we are about to see. Before Charles Darwin, I have told you that there are some evolutionary scientists like Lamarck, Jean Lamarck, who was a French zoologist, who had observed this evolution by natural selection. Actually, before Darwin, there wasn't enough evidence for natural selection, for that mechanism of natural selection. But we are saying that Lamarck is one of the earlier evolutionary biologists before Darwin who had observed that evolution takes place. He coined this Lamarck is the one who coined the term annelida and the term invertebrates. And his theory, which we call Lamarck's theory or Lamarckism or Lamarckian evolution, can be summarized in 4 factors. Factor number 1, that there is an internal vital force which causes all living organisms and their component parts to be continually increased. Number 2, the effect of environment and new needs. Environmental changes have an impact on organisms, causing them to change, leading to new needs. The new needs generate new structures and alter the habits of organisms. Number 3, idea number 3 in Lamarck's theory is the use and disuse of parts. Use and disuse of parts or entities. That as more organs or structures are used, they tend to develop better. And those which are less used or those which are disused tend to degenerate. The last idea in Lamarck's theory is inheritance of acquired traits. That characteristics adapted as a result of being used over and over again and continuing to develop better, those characteristics from those new or developed structures are passed on to the next generations, and that this is a continuous process. These variations are accumulated over generations, leading to the formation of a new species. Examples of support to Lamarck's theory. Some examples in organic evolution that support Lamarck's theory include the webbed toys of aquatic birds. Number 2, the evolution of giraffe. Example 3, the disappearance of limbs in snakes. Example 4, the flightless birds, the flatfishes, and the cave dwellers. But one other common example, not documented here, is the development of strong muscles in athletes. So according to Lamarck, heavyweight lifters and athletes who exercise their bodies to heavy activity tend to develop strong muscles. And according to Lamarck, such structures, as you continue to use them, they develop better, and they are inherited by the offspring in future generations. And this was one of the basis for criticism that was faced by Lamarck's theory. We know that strong muscles are not inherited by the offspring or progeny of athletes and heavyweight lifters. But Lamarck's theory suggests that such better developed organs or traits are transmissible to the offspring, which led to criticism and rejection of his theory. As already presented, the mechanism of evolution by natural selection has got a relationship with genetic variation. I've already told you that favorable variations have a survival value that they confer to their bearers, to the organisms that have them. But unfavorable variations have a disadvantage since they lead to elimination or death of an organism, of an individual. So the phenomenon of genetic variation is basic for organic evolution. It is upon that selective, it is upon this phenomenon of genetic variation that selective forces act for evolution to take place. The mechanism of evolution emphasizes on, number one, descent and genetic differences which can be inherited to the next generation. So the mechanism of evolution emphasizes descent and genetic differences or variations that can be inherited. Genetic change or genetic variation is attributed to the mechanisms such as genetic drift, migration, which includes emigration and immigration, mutation, and natural selection. So the mechanisms of change that are likely to result in evolution include genetic drift. That is one mechanism. The second mechanism is migration, either emigration or immigration. The third mechanism of change is mutation. And the fourth is natural selection. So each of these mechanisms, genetic drift, natural selection, migration, and mutation, confers some survival value. Actually, what we need to discuss here for these mechanisms, genetic drift, migration, mutation, and natural selection, is that they bring about genetic variation through changes in gene frequency or through changes in allele frequency. The changes in the gene frequency resulting from genetic drift, the changes in gene or allele frequency resulting from emigration or immigration, The changes in gene frequency resulting from mutation and those resulting from natural selection, where natural selection here could be disruptive selection, stabilizing selection, and directional selection. All of those changes, directional selection, disruptive selection, and stabilizing selection, lead to change in gene frequencies with the time. And the changes in the gene pool of any population that has experienced any of these mechanisms, genetic drift, migration, mutation, disruptive selection, or directional selection, or stabilizing selection, all these changes in gene frequency, changes in gene pool will result to evolution, will result to formation of a new species from a pre-existing species, which we call speciation. Okay, now we can briefly talk about this one by one. Mutation is a driving force for evolution. It can be a random change in the genetic composition, having an impact on the gene pool of a population. This alteration in the nature of DNA in one or more chromosomes produces new alleles, hence causing genetic variation. Natural selection acts by eliminating the less fit organisms and allowing the survival of the more fit individuals. It eliminates organisms with unfavorable variations and favors the survival of organisms with favorable variations. The second mechanism is natural selection. And this occurs when entities or organisms are left up to an environment where survival of the fittest is observed. These characteristics, when inherited in offspring, reproduce a population better suited to the changing environment. Traits from less fit entities or organisms less likely to be passed to the forthcoming generation. Natural selection, all the three forms of natural selection: disruptive selection, stabilizing selection, disruption, stabilizing, and direction. The significant selective force is the role of the environment. I request you members to read about the three forms of natural selection observed in populations: stabilizing selection and how it leads to formation of a new species from a pre-existing species. Read about disruptional or disruptive selection and directional selection, and how each one of them leads to formation of a new species. But also important to talk about selection is the concept of sexual selection. For example, the ability to attract and mate can also be seen as a measure of fitness, which we are calling here sexual selection. If you can attract and mate with as many females as possible, then the chances of producing more offspring and transmitting your characteristics to future generations will be high. And that is biological fitness. Those who are better adapted produce comparatively more offspring, passing on their genes at a higher rate than those who are poorly adapted. Migration is a mechanism which influences gene flow. Because as I've told you, emigration will lead to loss of some genes from one population to another, thereby changing or reducing the gene frequency. While immigration leads to an increase in allele frequency or gene frequency as individuals leave other populations to join this population. So migration of organisms may cause a cluster of them to move to a new geographical location. When the migratory entities or organisms interbreed with the newly introduced population, they result in the addition of new genes to the already existing gene pool as a result of the local population, thereby contributing to the gene flow. For instance, when pollen grains are blown by wind beyond the reach of the parent plant population, gene flow takes place. Some animals can be shifted away from a herd, compelling them to move to a new location, interbreed, and introduce new genes to the pool. Another mechanism of evolution is called genetic drift. This can take place when a small population moves to a new geographical location, establishing in a completely isolated area. For example, when a few fishes are introduced in a lake, the population evolves over time into a different one from the parent population. This process does not take place in densely populated areas. So genetic drift does not take place in large populations, but it occurs in small populations that have moved to a new location. And in the newly introduced location, in the new location, the population, that small population, will evolve over time into a different population, a population different from the parent. A particular group of entities, we are talking about development of a species, what we call speciation. And we are saying that a particular group of organisms sharing a number of traits with the ability to interbreed with one another, leading to the production of fertile offspring, is known as a species. So a species is a group of organisms sharing a number of traits and able to interbreed freely with one another to produce fertile offspring. And these individuals usually share the same gene pool. So speciation is the evolution of a species taking place when a specific population is isolated as a result of barriers, which barriers could be geographical barriers, ecological barriers, reproductive barriers, mechanical barriers. So I implore you also to read about isolating mechanisms. Read about reproductive isolation, ecological isolation, and geographical isolation. So you can have geographical isolation, reproductive isolation, ecological isolation. So individuals belonging to the same species or same gene pool could get separated by a geographical barrier, such as a river, such as a mountain. And with that isolation or separation, they fail to interbreed. And over time, each of them develops into a separate species. With the ecological isolation, still it could be due to maybe habitat isolation. For example, if members of the same species, one decides to live in the tree canopy and another one lives on the forest floor. Over time, these two groups that do not interact will evolve into separate species. And that is ecological isolation. But reproductive isolation could also be as a result of failure for groups of organisms belonging to the same species to interbreed or to mate with one another as a result of maybe some form of incompatibility. Some form of incompatibility. For example, if one group enters courtship or becomes reproductive ready to mate when another group is not yet ready, that incompatibility, if it continues to exist in the two groups of the same species over time, it will lead to reproductive isolation and an emergency or formation of two different species. So the phenomenon can also take place. I said the phenomenon of isolation can take place as a result of geographical barriers, but it can also take place with the development of reproductive barriers, such as anatomical hurdles that prevent mating of members of the same population. So that one is also likely to result in formation of new species. But I've also said that speciation can also form as a result of spatial differences and timing of sexual activity. That is known as reproductive isolation. So read about the isolating mechanisms and how those isolating mechanisms, mechanical isolation, ecological isolation, reproductive isolation, and geographical isolation, how do they lead to formation of new species? The next slide mainly talks about evidence for organic evolution, evidence for common descent, evidence for universal ancestry of all life on Earth. I'm very sorry, the color here, I did not know that it is dull. The color is dull. Maybe I'm going to see how to improve on this color later. But let me first talk over this. I'm going to talk through these slides. Evidence of common descent among living organisms. So, a variety of evidences as I've already shared with you, demonstrate that all life on Earth comes from a single ancestor. This forms an important part of evidence on which all evolutionary theory rests, demonstrating that evolution actually occurs and illustrating the processes that created Earth's biodiversity. It supports the modern evolutionary synthesis, the current scientific theory that explains how and why life changes over time. Evolutionary biologists document evidence of common descent all the way back to the last universal common ancestor by developing testable predictions, testing hypotheses, and constructing theories that illustrate and describe its causes. Comparison of the DNA genetic sequences of organisms has revealed that organisms that are phylogenetically close have a higher degree of DNA sequence similarity than organisms that are phylogenetically distant. The first evidence we are talking about is from genetics. One of the strongest evidence for common descent comes from gene sequences. Comparative gene analysis, comparative sequence analysis, examines the relationship between the DNA sequences of different species, producing several lines of evidence that confirm Darwin's original hypothesis of common descent. If the hypothesis of common descent is true, then species that share a common ancestor inherited that ancestor's DNA sequence, as well as mutations unique to that ancestor. More closely related species have a greater fraction of identical sequence and shared substitutions compared to more distantly related species. So this is an evidence of evolution based from genetics, and we call it comparative sequence analysis. It examines the relationship between the DNA sequences of different species. It produces several lines of evidence that confirm the theory of common descent. So if we are to go with that hypothesis of common descent, then since species sharing a common ancestor inherited the ancestor's DNA sequence plus the mutation is unique to that ancestor, then we conclude and say that where there is a greater fraction of identical DNA sequence and shared substitutions, then such species with a greater fraction of identical sequence are more closely related compared to species with a smaller fraction of identical sequences. The simplest and most powerful evidence is provided by phylogenetic reconstruction. Such reconstructions, especially when done using slowly evolving protein sequences, are often quite robust, quite strong, quite reliable, and can be used to reconstruct a great deal of the evolutionary history of modern organisms. These reconstructed phylogenies recapitulate the relationship established through morphological and biochemical studies. So this statement means that if we follow the phylogenetic reconstructions using DNA sequences or protein sequences, strong evidence is provided that the modern organisms, the present-day organisms, came from the same ancestor, and which is also supported by morphological and biochemical evidence. So some of these articles are actually articles written after carrying out a lot of research in laboratories, research in molecular genetics, or research in molecular biology. But the major point to note from this slide and from this evidence of genetics to support the theory of common descent is that comparative sequence analysis provides evidence that when the DNA sequences of different species is compared, we can obtain, or what we obtain is that closely related species have greater fraction of identical sequence, and distantly related species have a smaller fraction of identical DNA sequences. The second evidence to support common descent, to support natural selection and the theory of common descent, is known as paleontology. So when you are discussing these evidences, always emphasize that the evidence supports common descent. It supports the view that modern organisms share the same ancestor. Number two, demonstrate that the present-day organisms have evolved from that ancestor through a process known as descent with modification. Because I told you those are the two main ideas, the central tenets in Darwin's theory. The first tenet, the first idea is common ancestry, common descent. And the second idea is descent with modification. So as you're discussing how each of these evidences supports the theory of organic evolution, Darwin's theory, always labor to show how the evidence provides common descent, evidence for common descent, and how it also provides evidence for descent with modification. So paleontology is the study of fossils. Fossils are formed with the remains of plants or animals which get implanted in water or soil and are preserved for years together. So the footprints, the bones, the shells, the skeletons, the molds, those of organisms which lived in the past, they are the fossils that are compared with structures of present-day organisms in order to trace, in order to reconstruct the evolutionary history of an organism. So fossils were preserved in the form of hard structures of spine, exoskeleton, shells, bones, scales, feathers, teeth, and hair of previously dwelt organisms. Now, here is the evidence. Fossils are important for estimating when various lineages developed in geological time. As fossilization is an uncommon occurrence, usually requiring hard body parts and death near a site where sediments are being deposited, the fossil record only provides sparse and intermittent information about the evolution of life. So this means that not every living organism or its parts can be fossilized. And therefore, there are some gaps of the fossil evidence in explaining how the present-day organisms have evolved. Evidence of organisms prior to the development of hard body parts, such as shells, bones, and teeth, is especially scarce, but exists in the form of ancient microfossils, as well as impressions of various soft-bodied organisms. The third evidence, but of course, before we go to the third evidence, we note that this evidence of fossils is important because a number of fossils from extinct organisms, when compared to the present-day organisms, they show some similarities that could be used to show common descent. And also, some structures in present-day organisms, such as bones, teeth, have somehow become more complex compared to similar structures that are discovered from the fossil evidence, but retaining a number of similarities. Similarities in teeth, in feathers, similarities in bones, similarities in shells and other fossils. And that is an evidence of descent with modification. In other words, simpler structures gradually got modified over time into the present day complex structures in modern organisms. Another evidence is comparative anatomy. The comparative study of the anatomy of groups of animals shows structural features that are fundamentally similar, and we call these homologous structures, demonstrating phylogenetic and ancestral relationship with other organisms, most especially when compared with the fossils of ancient extinct organisms. So what we are saying is that different groups of animals show homology. Homology meaning similarity, fundamental similarity in structural features, such as the pentadactyl limb in mammals is a homologous structure, which is also found in the monkeys, found in the naked mole rat, found in birds, found in rodents, found in elephants, the same pentadactyl limb fundamentally or basically is the same in all those groups of animals. And that is an evidence for common descent or common ancestor, and also an evidence for descent with modification, because that pentadactyl limb has been modified into different structures that perform different functions. In the chimpanzee and monkeys, for example, you will find it being used for grasping or for climbing. In other mammals, you'll find it being used for digging holes, making burrows, and other uses. So, homologous structures are an evidence for common descent, as they have the same basic plan, and also an evidence of descent with modification, as the limb has been modified in different animals to perform different functions. It could also be used for flight in the bats. It has been modified into wings that are used for flight in the bats and the birds. Another evidence from comparative anatomy is vestigial structures, which we know that vestigial structures are structures that have degenerated. They have reduced in size. They are traces of former structures. But now these are now degenerated. They are much reduced in size as they have lost function in the surviving organisms over a period of time. So when the function of these structures or organisms is lost, they degenerate, they reduce in size, but the structures remain visible. And they provide an evidence for descent with modification. So we can compare the embryonic development and of different animals, and we will find that at some embryonic stages, some structures are present, but as the organism develops, maybe into an adult, from embryo stages to adult stages, some structures are lost, which confirms common descent. Further evidence is provided by geography or distribution studies. Distribution of plants and animals across the globe by geography provides evidence for evolution with common descent. When we look at the animals found, for example, in Africa, being similar to some animals on other continents like Europe, Eurasia, South America, or North America, we use the geographical theory of plate tectonics and continental drift to indicate that these widely separated plants and animals that show a lot of resemblances must have descended from the same ancestor. But the separation of these species arose from continental drift, where different land masses drifted apart from the originally single large landmass. And with this drift and geographical barriers that arose, such as the seas, the oceans, the mountains, the rivers and lakes, with those separations or geographical barriers, these species evolved along different lines, resulting in their differences. Otherwise, originally, they were similar species occupying the same landmass before the continental drift. We can obtain evidence for evolution also from present-day changes, the present-day evolutionary changes. For example, the resistance of bacteria to antibiotics, the resistance of ticks to acaricides, the resistance of malarial parasites, Plasmodium spp, its resistance to anti-malarial drugs such as chloroquine, quinine, Fancida, and other drugs. The resistance of those parasites to the drugs. Even the resistance of a number of herbs or weeds to the herbicides. The resistance of fungi to fungicides. All of those provide further evidence that evolution is contemporary. Evolution is an ongoing process in the natural world. Even if we look at the different strains of HIV that become resistant to different antiretroviral therapy. A number of HIV strains are evolving, becoming resistant to different lines of ART, antiretroviral therapy. And that is a challenge to science. It's a challenge to science, a challenge to research, that experimentations and trials must be an ongoing process since organisms keep on evolving and resisting the drugs, the chemicals that are used to kill them. So, scientists have shown these changes. They have demonstrated that evolution is an ongoing process, both in the laboratory and in the natural world. We can also use the evidence of selective breeding to show that evolution is still contemporary. Evolution is still an ongoing process. Selective breeding of plants, biotechnology is doing this. Selective breeding of animals, genetic engineering techniques that lead to development of new varieties, development of new new strains. All of this confirm that evolution is an ongoing process. So when we are looking at such evolution over a small period of time, like the one demonstrated or observed in the laboratory, over just a few minutes, within a few hours, within a few days, we find that the genetic material has changed. A genetic mutation has taken place. The gene frequency or allele frequency has changed within the lab environment, within hours. That evolution on a small scale is known as microevolution. But if we are looking at the evolution of the modern horses from the extinct horses, the evolution of the present day giraffes from their ancestors, the evolution of modern humans, Homo sapiens, from the early man, that evolution that occurs over a period of several years, evolution spanning over thousands or even millions of years, producing gradual changes in the anatomy, gradual changes in the physiology, gradual changes in the structures and the biochemistry of individuals, resulting in what we see today. If we follow such evolution that spans across thousands of years, that is macro evolution. That is macro evolution. Present day science of evolution is being expanded by techniques from molecular genetics and molecular studies of DNA and RNA to explain how evolution actually takes place, to explain the likely changes that might have taken place at a basic level, leading to descent with modification, as explained by Charles Darwin. And what we use is called DNA sequences, DNA sequencing, DNA sequencing. So comparison of DNA sequences, like we said, allows organisms to be grouped by sequence similarity. And the resulting phylogenetic trees are typically congruent with the traditional taxonomy. And they are often used to strengthen or correct taxonomic classifications. So taxonomic classifications have been updated. Taxonomy has been improved by what we call molecular genetics. And that new branch of taxonomy, where we use DNA sequencing to group organisms according to the sequence similarity of their DNAs. Formation of phylogenetic trees on the basis of sequence similarities. That science, which we call molecular taxonomy, agrees with the traditional taxonomy, with the way Carl Linnaeus classified plants. And what this molecular taxonomy helps, what DNA sequencing helps, is to indicate at a basic level, at a molecular level, at DNA level, how different organisms are genetically related. So this sequence comparison is considered a more robust process, a more robust evidence that shows that organisms placed in the same taxonomic grouping share common ancestry. So if we have members in the same species, it is likely that those have the same ancestor. Individuals in the same genus, it's more likely that they share a common ancestor. Individuals in the same family are more likely to have a common ancestor, at least at a certain period of time in the history of their evolution. They must have had the same ancestor. Individuals in the same order must have, at a certain point in time, the same ancestor. Individuals in the same class must have had, at a certain point in geological time, in the geological past, a common ancestor. Individuals in the same phylum must have had, at a certain point in the geological past, a common ancestor. So that is the essence of taxonomic evidence. It is the essence of DNA sequencing evidence. An example is that neutral human DNA sequences are approximately 1.2% divergent from those of the chimpanzee. The human DNA sequences are approximately 1.6% from those of the gorilla. The human DNA sequence is approximately 6.6% divergent from the DNA sequence of baboon. So what does this DNA sequencing show? Now, the level of divergence, the higher the level of divergence, the more distant the two species are. The closer or the smaller the level of divergence of DNA sequences of two species, the closer those two species are in evolutionary terms. Therefore, that information, that data indicates that a chimpanzee is more closely related to a human than the gorilla and baboon. But also, the gorilla is more genetically related to a human than a baboon. And also a baboon is more genetically related to a human than a monkey. So all of these are modern evidences, modern ways of showing genetic relatedness and evolution. So in terms of evolution from the common ancestor, among the humans and other apes, we may have the monkeys being more ancient, followed by baboons. Gorillas are recent, and the most recent, the closer to human evolution, are the chimpanzees. Now let us talk about comparative biochemistry, also known as proteomic evidence. Proteomic, sorry, it is proteomic. Proteomic evidence also supports the universal ancestry of life. Vital proteins such as the ribosome, another protein is DNA polymerase, and another protein, RNA polymerase, are found in everything, all living things, from the most primitive bacteria to the most complex mammals. So, proteins like DNA polymerase, RNA polymerase, and ribosomes occur in bacteria which are very ancient and also very small, but they have these proteins. But even the more complex mammals like the elephant and the humans also share the same proteins. And that is some evidence that right from bacteria to the complex mammals, there is some common ancestry. The core part of the protein is conserved across all lineages of life. All lineages of life have got that core part of these proteins, which is made up of amino acid sequence, what we call the primary structure of a protein. So that is a very strong evidence that all life forms have a common ancestry. We also see similarities between different lineages of extant organisms. We see similarities in DNA. These are known as biomolecules. Similarities in RNA, similarities in amino acids. For example, you will find that in all cells of eukaryotes, there are 20 different types of amino acids from which all proteins are derived, whether in plants, in mammals, in fish, in all eukaryotes. This is what you find. Same types of amino acids. The bilayer is similar, and this supports the theory of common descent. So we need to look at different molecules, the proteins, the vital proteins, the biomolecules, RNA, DNA, the lipid bilayer in the cell membrane, amino acids, all of those are similar across various groups. All the way from the smallest bacteria, the most primitive bacteria, to the most complex mammals living today, which indicates common ancestry or common descent. This slide indicates or shows, it emphasizes the genetic relationship, genetic relatedness of humans and chimpanzee by looking at chromosome 2 in humans. Remember, evidence shows that humans and the chimpanzees have evolved from a common ancestor. But what is that evidence? The evidence is found in chromosome 2, chromosome number 2 in humans. When you compare chromosome number 2 in all of the hominids, all hominids have 24 pairs of chromosomes, hominids. Apart from the humans who have 23 pairs, but human chromosome 2, human chromosome 2 corresponds to 2 ape chromosomes. So we can have chromosome 1 up to chromosome 23, which is the last chromosome in humans is the sex chromosome. The sex chromosome is the X and Y. They are heterosomes, they are dissimilar. But the rest of the 22 pairs of chromosomes in humans, from number 1 to number 22, they are similar and homologous. They are known as autosomes. They are known as autosomes. They occur in pairs. They occur in pairs, but they are similar. And the genes in them occupy the same locus in homologous chromosomes. So the only dissimilar chromosomes is chromosome, the last one, which we can call the sex chromosomes. The X is structurally different from Y and has different genetic composition, which is why you may find the males who are XY having some characteristics of maleness that are not found in females, but also the homogametic sex, the females, XX, also have traits characteristic of feminine or femaleness that are not found in males. So we are saying, if we number chromosomes of humans from number 1 up to number 22, chromosome 2, chromosome pair number 2 shows a lot of similarities. It corresponds to 2 chromosomes of the gorillas, chromosomes of the chimpanzee, and chromosomes of the baboons. The closest human relative, the chimpanzee, has near identical DNA sequence to human chromosome 2. But they are found in 2 separate chromosomes. The same is true of the more distant gorilla and orangutan. Orangutan is also a primate. So the distant relatives of humans, the gorillas, the baboons, the orangutan, the monkeys, the chromosome 2 is not near identical. It does not have near identical DNA sequencing or sequences to those of human chromosome 2. But that of the chimpanzee, they are very identical, near identical, almost the same DNA sequences in humans and in chimpanzee for human chromosome number 2, which is a strong evidence for genetic relatedness of humans and chimpanzee. We can obtain another evidence for common descent and descent for another evidence for descent with modification from comparative anatomy. The comparative study of the anatomy of groups of animals of plants reveals that certain structure features are basically similar. For example, the basic structure of all flowers in flowering plants, the angiosperms, or what we can call angiospermatophyta. The basic structure of all flowers contains sepals, petals, stigma, style, and ovary. It contains the sepals and petals, which are outside, and stigma, style, and ovary, which are inside. They are inside the petals. Yet, the size, color, number of parts, and specific structures of these flowers are different for each individual species. So if we are talking of the bean flower, the hibiscus flower, crotalaria, and other flowers, like the flowers of trees, like musizi tree, Mysopsis seminae, flowers of other trees, the mango tree. The flowers basically have these 5 parts: sepals, petals, stigma, style, and ovary. But when we are to talk about the color, the number of parts, that is the number of petals or sepals, the size of the flower, those are different for each individual species. We also have another evidence from comparative anatomy, and this is known as atavism. Atavism in biology refers to a modification of a biological structure. Modification of a biological structure whereby an ancestral genetic trait, a genetic trait that was in an ancestor reappears after having been lost through evolutionary change in previous generations. I am repeating. Atavism, an atavism, is a modification of a biological structure whereby an ancestral genetic trait reappears after having been lost through evolutionary change in previous generations. Atavisms can occur in several ways, one of which is when the genes for previously existing phenotypic features are preserved in DNA. And these genes become expressed through a mutation that either knocks out the dominant genes for the new traits or makes the already traits dominate the new one. So the point here is that we can have a trait, a phenotypic feature, or a trait that gets lost, that disappears during evolution. But its genes become preserved in the DNA of that organism, of that species. So later on, after some generations, when that trait or phenotypic feature had disappeared, a mutation occurs that causes the dominant genes of the new traits to get knocked out, And this mutation makes the genes for the already trait which had disappeared to become dominant again. And we will have the reappearance of an ancestral genetic trait that had become lost through evolutionary change in the previous generations. Some examples of this are provided by the following. The hind limbs in whales are an example of atavism. The reappearance of limbs in the limbless vertebrates. You never know. In the future, you will find snakes with walking limbs, with legs, and you'll find snakes walking instead of crawling. Another example, the back pair of flippers in dolphins. Number 4, the extra toes of the modern horse. Number 5, the human tails and extra nipples in humans. Number 5 or number 6, the re-evolution of sexuality from parthenogenesis in the mites. Number 7, teeth in chickens. Dew claws in dogs. The reappearance of wings on wingless stick insects and earwigs. The atavistic muscles in several birds and mammals, such as the beagle and the chiboa, and the extra toes in guinea pigs. All of these are examples found in scientific literature that show atavism. Atavism being the modification of a biological structure whereby an ancestral genetic trait reappears after having been lost through evolutionary change in previous generations. So we are also saying that atavisms are or were one thing they were once thought to be, and they actually are still, a great evidence of how genetic potential is retained, even if the phenotypic feature, the trait, had disappeared, had become lost through evolutionary change. There is some evidence of retention of the genetic potential of that trait, and it can reappear once a mutation occurs that leads to that trait, to that gene becoming dominant again and reappearing in subsequent generations. So I implore you also go and read about how comparative embryology, which I've already talked about, that you'll find different embryos from different groups of animals being similar. You'll find a tail possessing embryo in amphibians, a tail possessing embryo in mammals, in fish, in reptiles, even in birds, in the early stages of development, in the early days of life. Why is it that the embryos of humans, the embryos of elephants, the embryos of birds, the embryos of fish, the embryos of frogs, the embryos of different groups of animals are basically the same structurally? It is because those embryos have descended, they are for organisms that have descended from a common ancestor. And when the structures of embryos get modified to the present day organisms, to the present day humans, to the modern amphibians, to the modern elephants, to the modern rodents, to the modern fish, which are very different from those of embryonic stages that were similar. That is an evidence for descent with modification. So, ladies and gentlemen, it is important that we make further reading on the theory of Darwin, the theory of Lamarck, the isolating mechanisms, the types of natural selection, and the evidences for evolution. So my assessment will mainly revolve around Darwin's theory, Lamarck's theory, types of selection, and the mechanisms that lead to natural selection. That is the mechanism of genetic drift, the mechanism of emigration or immigration, the mechanism of selection, the mechanism of genetic drift. Those four: migration, genetic drift, and mutation. How do they lead to speciation, to the formation of a new species? I said they do that by bringing about change in the gene pool, change in allele frequency, resulting in the formation of new species. So read in general about the mechanisms of speciation, allopatric and allopatric speciation and sympatric speciation. That is allopatric speciation of species in different geographical locations, and speciation that occurs among organisms in the same geographical location. That is important. Also read about the isolating mechanisms. I will assess you about reproductive isolation, geographical isolation, and ecological isolation, habitat isolation, mechanical isolation. How do they lead to formation of new species? And finally, read about the evidences for the theory of evolution, evidences for natural selection, evidences for common ancestry, common descent, evidences for descent with modification. And these are comparative anatomy, comparative embryology, biogeography, taxonomy or classification studies, especially using DNA sequencing, and evidence from evidence from homologous structures, evidence from analogous structures, evidence from divergent evolution, evidence from convergent evolution. Those ones, you should read them as concepts or terms that you must know how to define and to give an example. I just wish you success as you go through this video, and I encourage you to do further reading, and you will pass this paper. It is not a hard paper. Thank you very much for listening to me. God bless you.